The Earth's atmosphere as it is today, the bedrock of all life, took millions of years and several intricate phenomena in the forming. The air we breathe and mostly take for granted is actually a complex cocktail of various elements. And therein hangs a question. What is the most abundant element in the atmosphere? If you answered oxygen, you'd be wrong. About 78% of the atmosphere by volume is nitrogen, represented by the letter N in the periodic table. This colorless, odorless, tasteless, inert gas, nitrogen, however, is an essential component of both plant and animal life. It's found in amino acids, the protein building blocks for muscles, skin and hair. In order for plants and animals to use it, the nitrogen goes through a whole cycle. The inert atmospheric nitrogen is deposited into the soil and surface waters with precipitation. Here, microorganisms break it down to ammonia. Other bacteria, called nitrifying bacteria, break ammonia into nitrites and nitrates, all of which are called reactive nitrogen. These are taken up by plants to form plant and animal proteins. Animal waste and dead plant and animal matter decompose and release ammonia and nitrates. The denitrifying microorganisms convert nitrates in wet soil back into gaseous nitrogen. In this natural cycle, the reactive nitrogen is reconverted into inert atmospheric nitrogen and the balance is maintained. The problem begins with human or anthropogenic interventions and activities. How exactly are humans generating the load of reactive nitrogen, and what is the impact? In India, 17 million tons is the nitrogen component of the total fertilizer used annually. Of this, only 33% is used by grain crops in the form of nitrates. The crop yield for every unit of nitrogen used is called nitrogen use efficiency or NUE. The rest, reactive nitrates, seeps into land, devitalizing soil and progressively depressing its productivity while contaminating surface and groundwater sources. Agricultural nitrogen is also responsible for the greenhouse gas nitrous oxide N2O, 300 times more toxic than the popular villain carbon dioxide. As air pollutants, other forms of reactive nitrogen like ammonia and nitrogen oxides are far more serious health concerns than generally realized.
Up to half of the particulates matter, which is what we call PM 2.5, the coating on it is because of particles of nitrates or NOx. So, half of PM 2.5 health effects or adverse effects are due to deposition of nitrates. Water contamination or even vegetables, when they bioaccumulate a lot of unnecessary nitrates in them, they can cause gastrointestinal disorders. Small increase in nitrous oxide can cause more serious global warming and ozone hole effects than large increase in carbon dioxide, and yet in the name of climate change we only talk of carbon, nobody even hears about nitrogen.
In India, industrial and organic sewage and solid waste from humans and animal husbandry follow agriculture in generating pollutants from oxides of nitrogen. The third source is vehicular pollution and combustion.
Many of them come from fuel emissions, coal burning, diesel burning, petrol, residue burning, municipal waste burning, crop burning, stubble burning. So, all kinds of burning produces NOx compounds, which also add to the reactive nitrogen in the air.
With nitrogen fast becoming a grave offender in global climate change, the Nitrogen Group was set up as an NGO in 2006 to scientifically look into the negative effects and possible solutions. Their nitrogen assessment discovered that we produce reactive nitrogen far in excess of what the food chain can utilize. And this overload, released into the land, water and air as a pollutant, cannot be naturally neutralized.
The natural process of neutralizing reactive nitrogen into less reactive or unreactive nitrogen gas, the denitrification, is also very limited. So, nature's capacity to neutralize reactive nitrogen back into unreactive nitrogen is limited. So, this means that any anthropogenic reactive nitrogen generation should be very, very little, only as much as we can consume, but that is what is not happening.
That is the concern, is to reduce the reactive nitrogen, and one of the ways of doing that is to reduce chemical fertilizer consumption.
To tackle the issue of nitrogen, India's Department of Biotechnology (DBT) entered in a bilateral agreement with the Biotechnological and Environmental Research Councils of UK, BBSRC and NERC, now merged under the UK Research and Innovation (UKRI). They set up 4 Newton Bhabha joint virtual nitrogen centres to develop and test practical solutions that can tackle aspects of the problem effectively. The Indo-UK Nitrogen Fixation Centre, IUNFC, a consortium of 7 institutions, with collaboration from Oxford, has the theme of improving biological nitrogen fixation.
Biological nitrogen fixation is the second most important biological process on this planet Earth, photosynthesis being the first.
Plants cannot convert the inert nitrogen to usable compounds themselves. In leguminous plants like chickpea, natural nitrogen fixation occurs when bacteria like rhizobium, present in their root nodules, convert inert and unavailable nitrogen in the air to ammonia, which plants can readily use. The man-made method is the Haber-Bosch process, which converts inert nitrogen into usable ammonia and nitrates at high temperatures in the presence of catalysts. This can produce nitrates and ammonia used as chemical fertilizers on an industrial scale.
So if you look back around, say, 50 years ago, the amount of chemical nitrogen fixation by Haber-Bosch process was only about 50 million tons. And biological nitrogen fixation was something around 140 million tons, so 3 times. But then gradually what happened was the things have got reversed. Today, the chemical nitrogen fixation by Haber-Bosch process contributes something about 165 million tons. It's about 150, but likely to increase to 165. Biological nitrogen fixation has come down from 140 to 100 and even going to come back to about 90 million tons. So that is the major concern. So we need to improve biological nitrogen fixation by all means.
Biological fixation has been traditionally used to increase nitrogen fixation through legume intercropping with wheat and rice. As a part of DBT UK Collaborative VNC program, a team of scientists at the Indian Institute of Soil Science, Bhopal, are working on increasing levels of biological nitrogen fixation in legumes.
Legumes are the basis for sustaining agriculture production in the absence of chemical fertilizers. There is no other way. Either you do it through legumes or do it through farmyard manure, vermicompost and others, composting, which requires a lot of organic resources which have to be recycled and then composted.
The team is currently working on the leguminous plant pigeon pea or tur dal that are grown extensively all across India.
You see the structure of nodule here, okay? And this one, now this is already dead. You know, so what happens is when the nodules will become, they've already given up all its nitrogen.
The idea is to keep the yield high while increasing the number and quality of rhizobia strains in the legumes to maximize nitrogen fixation from air and soil.
Let me tell you that the rhizobial populations in soil are very low, and that's why we need to inoculate the plants with these rhizobia. So these are called as biofertilizers or biological preparations by which we can make the plant fix more nitrogen. So these strains have been screened across 3 soil types, and about a dozen strains out of these have now been identified as the best strains. These have now been sent for whole genome sequencing, and these are now being tested in the fields.
Their work also includes identifying bacteria and introducing enzymes in the rhizobium that effectively solubilize phosphates in the soil. Phosphates are critical for optimizing nitrogen fixation. making these strains of bacteria doubly beneficial. Further, the rhizobia-hosting legume crop is also treated as a green manure for the intercropped rice plants.
These rhizobia are there in the rice plant ecosystem because this is a rotation of a legume with a rice plant. So those rhizobia also infect rice plants and then act as endophytes. So they go inside, and then some of them also fix nitrogen. So trying to find out what are those associations where the endophytes are benefiting the rice most.
The growing awareness for sustainable organic farming is reflected in farming trends in the past couple of decades. Efficient use of nitrogen is a natural progress in this direction. Another DBT UK joint virtual nitrogen centre, Newton Bhabha Virtual Centre on Nitrogen Efficiency of Whole Cropping Systems, or NEWS, tackles nitrogen use efficiency at another depth.
When these things come to just before you start evaluation of these plants.
They point out that unutilized reactive nitrogen, whether chemical or organic, is a problem on our hands. NEWS approached the solution from the plant end by examining its structure and the entire cropping system. They studied one crop, rice. Rice, they reasoned, not only feeds half the world, but is also the biggest consumer of urea, which accounts for most of the agricultural nitrogenous emission.
That is why we deliberately chose rice as the focus, not only because it is an Indian crop, not only because rice is a very good model crop whose genome is sequenced and it is a very popular system after Arabidopsis even in basic biology, but also because we have this information available about rice consuming the maximum fertilizer in India.
India alone has more than 10,000 varieties of rice. And the first challenge lay in identifying rice varieties that could be in the race for optimizing nitrogen use. One method was to identify the varieties on the basis of their phenotype or physical features.
Phenotype is a visual identification of a trait or a feature. In this case, nitrogen use efficiency as a feature. If plants have certain kind of structural features that help them to use the fertilizer supplied to them better, and if we are able to find those features, whether it is a long root or branched root or a time of flowering or germination itself, we hope to be able to reduce the total number of parameters which are usually measured in every crop by agricultural scientists.
The team took note of what was known about certain varieties of rice and to see what could be quickly and easily established about a plant feature that connected to nitrogen uptake.
We have analyzed their germination profiles in simple lab-based plate experiments. We showed that different varieties of rice seed has different rates of germination.
They found that slow germinating varieties had longer crop duration and higher yield, even with lower applications of nitrogen, indicating their higher nitrogen use efficiency compared to fast germinating or short duration varieties. The phenotype is, to some extent, linked to the genetic traits of the variety. So, the next step for the crop improvement and nitrogen use efficiency was screening these rice varieties for genotype.
India Science is an internet-based science TV channel initiated by the Department of Science and Technology, Government of India, implemented and managed by Vigyan Prasar. India Science on the URL www.indiascience.in is a bilingual channel with content in Hindi and English. It can be accessed on any internet-enabled device. This video platform is dedicated to science and technology knowledge dissemination with a strong commitment to spreading scientific awareness, especially with Indian perspectives, ethos and cultural context. The India Science mobile app can be downloaded from the Google Play Store and Apple Store.
What if India's greatest challenges, food security, weather prediction, farming livelihood, healthcare for all, education, law and order, could be solved not just with hard work and dedication, but with innovation, new age technologies, and the mighty power of computation. Watch the transformation only on India at IT Revolution.
Genotype is the complete genetic identity of a life form, which is mapped by genome sequencing. Out of the innumerable genetic traits, they were looking for a few specific ones.
We have shortlisted a few hundreds which we think are important for contributors to nitrogen use efficiency. Out of that, we were able to identify overexpressing transgenic lines for 40 genes. Those are the lines that we are growing in the greenhouse to see which of these 40 genes line by line contributes to nitrogen use efficiency. Ultimately, you have to be able to bring the phenotype and genotype together, bring classical genetics, reverse genetics and modern breeding techniques together. Then for each of the gene you can develop genetic markers, and you can use genetic markers to do marker assisted selections or marker assisted breeding.
By combining multiple approaches, the news team is already doing field trials for varieties they've identified and expects reliable results in 3 years, where conventional methods could have taken about 10 times that period. These are results that the DBT UK VNC project expects can be replicated with some tweaking with other crops like corn, oilseeds or wheat. Wheat, rice and other crops are also where another joint virtual centre, Cambridge India Network for Translational Work in Nitrogen, Cintron, has demonstrated how farmers can immediately benefit from and achieve the common goal of nitrogen use efficiency. Indian farmers are usually inclined to use excessive urea to get lush green leaves, which they interpret as a sign of healthy crop.
Landowner has used 180 kg of urea per acre here, due to which the crop has been destroyed.
This not only results in high levels of unutilized reactive nitrogen, but also leads to foliage overgrowth, disease and poorer yield.
See, all seeds are black here. We lost all of the crop varieties. After seeing this, farmers feel that they have used too much of fertilizer here that leads to the huge losses.
Using optical sensors, chlorophyll meters and other tools, the Punjab Agricultural University developed a leaf color chart to determine the optimal nitrogen for different crops.
This is the solution to our problem. According to this chart, you use the fertilizer. If we use fertilizer according to the need, then the attack by insects will be reduced. Diseases will also be less. Problem of black seeds will also get solved.
The right shade of green was correlated with the quantum of nitrogen to be applied or not applied by comparing it with the leaf color chart for a particular crop.
This tool is so smart tool, it take care of nitrogen supply from the irrigation water, biological nitrogen fixation, or seasonal variability, or release of nitrogen from the organic manures. It takes care of supply, nitrogen supply from all the sources, and finally it gives the decision. That decision, the instant decision. While standing in the field, farmer can immediately know whether he shall apply or he shall not apply, and how much to apply. So this is the beauty of this tool.
Using better yield as leverage, they persuaded farmers in this field study at village Basiyan to use it to determine how much urea should go into a field.
Do not use fertilizer in stagnant water. Use it in running water. This will solve the problem.
The result is speaking.
Our experiences with rice and wheat have shown that in case of rice there is a saving of 75 kilo per acre of urea, and in case of wheat there is a saving of 50 kg urea per acre. So accordingly, as per the area of rice and wheat in Punjab, we have calculated that there is a saving of 750 crores in total, and out of this 500 crore is the saving of the government and 250 crore is the saving of the farmers.
Leaf color chart is very cheap tool. Surely use it. I have used 180 kg of fertilizer per acre as per my own consideration. And as per the leaf color chart, I have used 75 kg of fertilizer per acre. By using leaf color chart, I have saved 105 kg of fertilizer. When one farmer told the other that I have saved 105 kg fertilizer per acre, then what was the reaction? Almost 99% of farmers have changed and using LCC because they have incurred huge losses.
What are the implications of this result for the use of the leaf color chart?
India Science is an internet-based science TV channel initiated by the Department of Science and Technology, Government of India, implemented and managed by Vigyan Prasar. India Science on the URL www.indiascience.in is a bilingual channel with content in Hindi and English. It can be accessed on any internet-enabled device. This video platform is dedicated to science and technology knowledge dissemination with a strong commitment to spreading scientific awareness, especially with Indian perspectives, ethos and cultural context. The India Science mobile app can be downloaded from the Google Play Store and Apple Store.
What if India's greatest challenges, food security, weather prediction, farming livelihood, healthcare for all, education, law and order, could be solved not just with hard work and dedication, but with innovation, new age technologies, and the mighty power of computation. Watch the transformation only on India at IT Revolution.
The ease and reliability of the leaf color chart method makes it a model candidate for replication in crops all over India, UK and other countries. While the DBT UK collaborative VNCs are successfully seeking solutions and showing the way to mitigate and neutralize reactive nitrogen in the area of the biggest source, agriculture. It is in our best interest to take swift and concurrent steps in containing all areas of reactive nitrogen. Investing in parallel research and making swift decisions in managing and containing solid waste, industrial and vehicular pollution is of the essence. The only constant in finding better ways to respond to our environment is reacting in good time to change. Please mail your feedback on feedback@indiascience.in. You can also write to us on Vigyan Prasar, A50 Institutional Area, Sector 62, Noida, Uttar Pradesh, 201309. For more information about our programs, please visit our website www.vigyanprasar.gov.in.